My name is Tiffanie Nelson and I'm working for the Australian Institute of Marine Science. I'm working on a project that is looking at the effect of the saltwater intrusion out here in Kakadu. The saltwater moves across out of the river on top of the floodplains into the freshwater habitat during the monsoon period, which is from November to March, and this changes the habitat structure. So when you get a freshwater habitat and you get saltwater intruding, the vegetation dies back, and so you get a complete change in the vegetation structure, and this affects the type of animals that can live in the habitat. Small animals and bacterial cells are really important in the ecosystem, and particularly the soil environment, because they're so abundant and diverse. They do a lot of jobs that make plants able to grow in this environment, which other animals are reliant on. So they cycle a lot of nutrients and they provide things like nitrogen, which makes the plants able to grow. So what we're trying to understand is how the saltwater that comes out of the river onto the floodplains affects the community in the soil, so the small animals and the single-celled organisms. The project that I'm working on is using a novel technique which uses genomics. So we take soil samples in the field environment and we take them back to the lab and we extract the DNA from the sample. So the DNA exists in all types of cells, so the small animals that live in the soil and then the single-celled organisms that are in the soil as well. So we take the DNA out and we copy using a complicated chemical technique that's called PCR. We copy the DNA and then we can analyse it and read the DNA, and that will tell us what type of species is in the soil. This is a much quicker technique than what used to happen, was that you would actually have to count all the individual cells and small animals that were in the soil, and this was very time consuming and often you would have difficulty identifying the animals, and so this way we can get a really quick snapshot of what's in the soil sample with a lot less time. From the study we've just done, we've got preliminary results that say that there's about 1,000 different types of bacterial species in 1 gram of soil. So we're getting also an understanding of the diversity of bacteria that are out here in these floodplain soils. This research will give a greater understanding to the biodiversity that is in Kakadu, and it's using a novel technique, the genomics technique. In the future we'll be able to use that as a tool, and we'll know if it's a viable tool to use to understand more about the biodiversity of floodplains or soil systems, and also to identify sites where saltwater intrusion is occurring.